Okay, let's get started with today's discussion on hierarchical clustering and dimensionality reduction. So we'll be continuing our discussion on unsupervised learning techniques today, and we'll see a new method for clustering, hierarchical clustering, and then we'll look at a new task called dimensionality reduction. Okay, a couple of announcements. Homework 4 due Friday. Don't forget about this one. For the discussion sections on tomorrow afternoon, tomorrow evening, the topic is going to be a live coding session on ensemble methods. So the TAs will walk through, what it actually looks like to implement a few of these ensembling methods, particularly bagging and random forests. So you get to see what it looks like to actually implement these in practice, and you'll get to ask questions. As always, it's a great idea to come prepared to these discussion sections with any questions you might have. So anything about ensembles that's particularly confusing, tomorrow's discussion is a good time to have those answered. And also note there's no class on Monday since it's Memorial Day, so everyone gets to take a short break. Okay, so to start off with, we're going to talk about k-means plus, small variation on the k-means algorithm. And then after that, as promised, we'll talk about hierarchical clustering and dimensionality reduction. Okay, so one of the main problems with k-means that we've seen so far is that it can get stuck in local minima. It's very sensitive to how you initialize these cluster centroids. So notice here in this graph, just by chance, when I initialize my 3 centroids for k-means, they're all very close to one another and all start up in this top left cluster. So the black points are my initial centroids, which again we choose by just picking random data points. So we kind of got unlucky. Our clusters at the initialization are all very close to one another. If we do 3 iterations of k-means, what you should notice is that iteration 2 and iteration 3 are exactly the same. So my cluster label assignments don't change after the second iteration. And so I've actually converged. But unfortunately, just because of bad luck, I've converged to a local minimum, where the squared error, which again is kind of the total distance between every data point and its corresponding cluster centroid, converges to a pretty high value. So we get stuck in a local minimum because of our bad initialization. One strategy that we can kind of employ to avoid this local minimum problem is to instead initialize our clusters in a bit of a smarter way. So rather than just randomly picking a few data points as our cluster centroids, we're going to do something a little bit smarter here. And the idea is we want cluster centroids at initialization to be far apart from one another. Intuitively, if we have 2 data points that are nearby, well, they're probably in the same cluster. And so ideally, we would like our centroids to be further apart, at least at initialization. So let's look at an example. So here we've got some unlabeled data that we'd like to cluster. If we get unlucky, our 2 centroids are just randomly selected data points that are going to be close to one another. In this case, they live in kind of the same cluster. But if we instead get lucky at initialization, well, we might pick our cluster centroids that are, pretty close to what they actually should be kind of at convergence of our algorithm. So if we're smart about initialization, we can save ourselves sort of a lot of computation and can solve that problem of getting stuck in local minima. So one way to do this is through an algorithm called k-means plus. And the way this works is that to initialize our k clusters, so we're choosing k, this capital letter k here is the number of clusters we're trying to fit. The first centroid we pick, C sub 1, is just randomly selected from the data. So we pick a random data point, and that's our first centroid. Now for the remaining k-1 centroids, we're going to pick these not randomly, but rather in a kind of adaptive fashion. Okay, so to pick the centroid C sub k, little k here, we're going to look at every data point that we have not yet chosen as a centroid. So we look at every single data point. And for each of these data points, we're going to compute a distance, d sub I, which is the squared Euclidean distance from that data point, x sub I, to the nearest centroid that we've already chosen. So if I've picked only the first centroid so far, I compute the distance between all of my data and that first centroid, and those distances are d sub I, so 1 distance per data point. And then to choose my kth centroid, I'm going to randomly select a data point, but rather than choosing them kind of randomly at uniform, I'm going to set each data point to have a probability of being chosen. So the probability of choosing the data point x sub I as my next centroid is going to be proportional to this distance d sub I. So if a data point is really close to my first centroid, the distance is going to be small, right? And so the probability of choosing that data point is also going to be small. If that distance is really large, if I have a data point far away from the cluster centroid I have already, well, then it's going to be chosen with a higher probability. Okay, and then we just iterate for each of our remaining k-1 centroids. We do this procedure. So this is just an initialization strategy. It's just a way of picking our initial centroids. And after initialization, we just proceed with k-means as usual, updating in this iterative fashion. The time complexity of doing this initialization is a little bit expensive. So it's in the order of n times d times capital K. So it's linear in the number of data points we have. So if we have a huge data set, this is going to be kind of expensive. But the nice thing is you only need to do this once at initialization, and you don't need to do this initialization over and over again when you're running k-means. So overall, k-means, fairly simple heuristic strategy for choosing our centroids such that we encourage them to end up far apart at initialization. Okay, let's see an example. So here I've got some unlabeled data, clearly got 3 clusters in this data set. And I'm not running k-means, I'm just randomly initializing the centroids. So in this slide, I just pick 3 data points at random from my data set to be the initial cluster centroids. And those are the red dots you see here. What you should notice in that all of these graphs, our cluster centroids often end up pretty close to one another. So up here, they're all in the first blob. Here we've got 2 in the first blob and 1 down here. Here we've got 2 in the middle blob. Here we get a pretty good one, but we just happen to get lucky here. So for 3 out of our 4 random initializations that we're looking at, we have a pretty bad start. K-means needs to do a lot of work to move these centroids to their correct positions. In contrast, I do this k-means plus plus initialization. What you see here is that these are all pretty good at initialization. I haven't even run k-means yet, and I already get pretty good cluster centroids. So in this first one, for example, I get 1 dot per centroid as my initial, 1 dot per cluster as my initial centroids. Same for these other plots. This one we get a little bit unlucky, but at least it's better on average than random initialization. So you might be wondering, why shouldn't we just initialize our clusters by picking the furthest data point in my data set from the existing centroids? We did this kind of strange probabilistic thing where we choose data points randomly with probability proportional to those distances. So let me ask you guys, why would it be a bad idea for me to just pick the furthest data point from the existing centroids? Yeah. Yeah, exactly. So the word I was looking here is outlier. So imagine you have, clusters that look like this, but you've got some data point way out here in the middle of nowhere. It's an outlier. Well, you probably don't want to initialize your cluster centroid as an outlier data point if it's going to be way far from the rest of your data. So by choosing them in this probabilistic fashion, you're more likely to not choose outliers while still encouraging this kind of separate behavior for your clusters. Okay, any questions on k-means plus plus? Yeah. Yeah, so essentially because it's probabilistic, right? Unless it's an extreme outlier, well, there's going to be some probability assigned to non-outlier data points here. Oftentimes you scale your data too, so that even if it is an outlier, it isn't going to have 99.99% chance of being chosen. So yeah, oftentimes this probabilistic thing will let you get around that problem in that way. And also recall that we run k-means often for multiple initializations. So even if you happen to choose an outlier on a particular instantiation of this, if you rerun this, you probably won't choose it again just because it's probabilistic here. Okay, other questions on k-means? Yeah. So in these examples, I'm only looking at 2 features. My data sets just have 2 features. But this all works for an arbitrary number of features. Just a lot harder to visualize in 1,000 dimensions, yeah. Yes, so you should use all your features when you're running k-means, unless you have a good reason to ignore 1 or 2 of them, yeah. Yeah. Yep, that's right. Yeah, we always choose our initial centroids as data points because, well, we have no better way, really. Okay, so in homework 5, which hasn't been put out yet, you'll actually implementing k-means plus plus, so that pseudocode I showed should be pretty helpful. Okay, let's move on and introduce a new topic here called hierarchical clustering. So this is a new set of methods that we haven't seen before, where we're going to be clustering data in a kind of top-down or bottom-up fashion. And the general idea behind hierarchical clustering is that rather than kind of developing a single clustering of our data, we're going to build a tree of clusters. And really this is mostly useful for sort of exploratory data analysis when you have small data sets. And this hierarchical clustering is really useful when you're trying to understand your data set and understand the relationships between particular data points. So for an instance of what you might do with hierarchical clustering is you have, say, a bunch of fruits, and you want to sort of group these fruits based on how similar each particular one is. So peaches and oranges, according to this tree at least, are quite similar to one another, so they kind of get clustered at a low level in this tree. Well, apples then are similar to both peaches and oranges, so they get clustered with this kind of level 2 cluster, if you like. Pears are similar to all of these guys, so they get grouped together once again. And kind of going further and further in this tree, we kind of group clusters of clusters together until eventually everything kind of reaches a root node here. Okay, so another example of hierarchical clustering that you might have seen are these kind of phylogenetic trees, as they're called, where rather than grouping together fruits, we're grouping together just different species of, say, bacteria or different kind of creatures. So these trees of life are all examples of hierarchical clustering. And we'd like to develop some kind of algorithmic automated way of doing this on unsupervised data. There's really two high-level approaches for doing hierarchical clustering here. The first is called agglomerative clustering, or bottom-up, a bit of a mouthful. But the idea is you start by merging individual data points into clusters, and then you can merge your clusters into kind of bigger clusters, and then bigger clusters, and so on and so forth, until all your points are in a single cluster. And we'll see plenty of examples of doing this. There's also divisive or top-down clustering, where you start with all your data and kind of break it down into smaller clusters. We won't be looking at this in the lecture because it's not really used all that often in practice, and so we'll focus on this agglomerative clustering setup. Okay, to do agglomerative clustering, we're going to need a notion of a distance between clusters. So far in the class, we've seen lots of distances between individual data points. And so we're going to need to kind of generalize these in order to define a distance between clusters of data points. So throughout, we're going to use the Euclidean distance as our distance measure between individual data points. But we could use other distance metrics here if we want to. So the first method we're going to use to define distances between individual clusters of data points. So I have a set of data and another set of data. How far away are they? What I'm going to do is define their distance as the distance between the two data points in these clusters that are the closest to one another. We'll see a visualization of this in a sec. The second way I can define a distance between two clusters is I can define their distance to be the distance between the maximally far away pair of points in those clusters. And the third method we can look at is the average, where we look at the distance between the centroids of these clusters. Okay, so visually, this single link distance method is going to compute a distance between the cluster of the yellow points and the cluster corresponding to the blue points. So for the single link, or nearest neighbor distance, the distance between these two point clouds is just the distance between this yellow point and this blue point, because they are the 2 points in the different clusters that are closest to one another. And I can use this distance as kind of a sort of heuristic for how far away these 2 point clouds are from one another. The second metric here, this complete link metric, or furthest neighbor metric, essentially computes the distance between the yellow cluster and the blue cluster, not as the distance between the closest points, but rather as the distance between the furthest points. So here, this yellow point is the furthest yellow point from the blue cluster over here. So we'll use this distance as the distance between the clusters. And then the third metric we could look at is this average link metric, where we look at the distance between the centroids of these two clusters. So overall, we've got three different ways of writing down distances between sort of sets of points here. Each one of these distances is going to give us qualitatively very different clusters in our hierarchical clustering algorithms, and so we'll see examples of sort of what effects this distance choice has. Okay, let's look at an example. Really, I think the example drives home what we're doing here and how this works. So here we're going to focus on this single link or nearest neighbor distance in this example. So say I've got some unlabeled data. Each one of these is green points is a data point. What we're going to do is we're going to initialize our algorithm by creating a separate cluster for every data point. So here I've got n clusters, one data point per cluster. What I'm going to do is I'm going to compute the pairwise distance between every single one of my data points. So how far away is each data point from every other data point? And we're going to store these and reuse them later for computational reasons. But whatever the closest pair of data points is, according to this distance, we're going to merge into a single cluster. So these two data points, these two green guys, are actually the two closest data points in this data set. So what I do is I merge them together. So I took my two data points and created sort of a cluster out of them. Now, what I've done on the right-hand side is I'm starting to draw what's called a dendrogram. And this dendrogram is a visualization of my hierarchical clustering. So these two data points correspond to these two guys, and I draw a link between them because I've clustered them together. The sort of height of this link is going to represent the distance between these two data points, and so higher height is going to represent sort of dissimilarity, if you like. So further distances mean further heights in this diagram. Okay, so I've done my first step. I've created one cluster. Now what I need to do is look at the distance between all of my data points, but rather than computing distances between, say, this green point and this individual red point, I'm going to compute distances between this green point and the whole cluster of red points. So we already saw there's a couple of ways we can compute distances between two different clusters. And here again, we're using that single linkage or nearest neighbor method. And if I do that, well, it turns out that these 2 points down here were the closest two data points. So I'm going to cluster those guys together into an orange cluster now. And here, note the height of this link I'm drawing as higher than the height of the previous one because it has a larger distance. Now what I can do is repeat this procedure, compute the distance between every individual cluster that I have. And what happens is this data point is now closest to the red cluster. So I group it together and create a larger cluster. And again, I draw a link between this cluster now and this individual data point. And then I just iterate this procedure until all of my data points are in a single cluster. So if I do this for very many iterations, I'll eventually end up with kind of 3 clusters in my data. So these guys all get grouped together, these guys and these guys all get grouped together. And again, I'm drawing links at every stage of this procedure. Okay, if I do one more step, well, these 2 clusters are the closest to one another, so I link them together and create a new cluster. And then at the very end of the day, well, I only have 2 clusters left, so of course I have to group these together, and I've put all of my data into a single very large cluster. That's sort of the root of my dendrogram. So in all, what we've done is, again, we've started out with all of our data in individual clusters, and slowly, step by step, merge them together into larger and larger clusters. In practice, you often stop doing this procedure once you've clustered all of your data into a single cluster, but there are other ways we could decide to stop this as well. So for example, if you know that there's 3 clusters in your data set, you can continue this procedure until you've only got 3 clusters. Or you might say stop after a certain distance threshold is met. So what I mean by this is if I don't know how many clusters I want to have in my data, I might say, okay, I don't want to group together clusters if their distance is above some prefixed threshold. In practice, what this actually means we're doing is we're taking our dendrogram and kind of cutting it. So again, the y-axis on this dendrogram represents distance between individual clusters. And so if we kind of chop our tree at some pre-specified distance level, we'll create clusters whose distance is below that threshold. So here we've got these orange points corresponding to this cluster here, the single data point corresponding to this single data point, and so on and so forth. So this is the main idea behind hierarchical clustering. Overall, relatively simple and sort of heuristic method for doing clustering. Okay, in sklearn, agglomerate clustering is of course implemented. There's a number of hyperparameters that you can tune and play around with. So the number of clusters here is essentially when we stop doing this clustering, it's the maximum number of clusters that we would like to set up. So you can either leave this as none if you want to group everything into a single cluster at the end of the day, or you can set this as some threshold. You can also choose what's called the linkage, which is again this distance function between individual clusters that we had introduced before. Okay, here's an example of what hierarchical clustering looks like on a real data set. So here we're looking at the iris data set, which again is a collection of flowers and various sort of features about those flowers, where all of the colors in this tree that I've created indicate their true labels. Note here that I'm not using the labels at all to do this clustering. This is a purely unsupervised method. It doesn't take into account the labels of these flowers. But using this hierarchical clustering method, we see that it actually successfully groups together pretty much all of the blue flowers with one another, all of the green flowers with one another, and there's a bit of sort of mixing down here, but most of the red flowers get separated as well. So this is cool because it tells us how kind of similar individual data points are in our data set. Okay, so any questions on hierarchical clustering so far? Yeah. So it is, conceptually, it's the distance between the clusters. So here I've got 3 clusters in this example, right? And I'm trying to decide, okay, how should I merge these guys? What I need to do is compute the distance between the red cluster and the blue cluster, between the red cluster and the orange cluster, and same thing for the remaining pairings, right? The way we compute distances between clusters is kind of up to you. It's the distance function that you choose. So if you use this single linkage or nearest neighbor clustering, distance, the way you compute distances between these clusters is it's the distance between the nearest points in those 2 clusters. So for example, the distance between the red cluster and the blue cluster might be the distance between this individual red point and this individual blue point. You could choose this to be the maximum distance, as we saw, or the centroid distance as well. Yeah, there's another question. Maybe it was answered. Okay, cool. Yeah, that's right. So the way you measure distance is exactly a hyperparameter of this method. So as we saw in sklearn, you can specify this distance when you set up this hierarchical clustering. And we'll see some examples of what effect different distances have in just a second. Yeah. Yeah, so you can think of, before we do any clustering, you can think of individual data points as already being in clusters where it's just a cluster by itself. So very lonely clusters at the beginning. We do one step where we maybe create a cluster with 2 data points, but most of my data points are in individual clusters. Well, we can compute the distance between this red cluster and individual data points because we can think of them as clusters themselves. And then you can use nearest neighbor or furthest neighbor or average. Yeah. Yeah, another question. No, so when you initialize, every point is in its own cluster, right? There's no choice going on here. And the first clustering we do is we look at the 2 data points that are closest to one another. So whatever 2 data points are closest, we're going to merge those into a single cluster. Yeah. Okay. Well, this is a purely unsupervised method, so we don't have labels for any of these data points. So all we know is there's some points out in space, and our assumption here is that if they're nearby in data space, if they have similar features, well, they're similar data points. We should group them together. So it's kind of limited in the sense that, okay, we might go out and collect more features for our data points that tell us, oh no, these guys are really supposed to be far apart, but based on the data we have, this is kind of what we can do with them. Yeah. Okay, let's look at an example of what effect choosing different distance metrics has on the shape of your clusters. So let's focus on this first row up here just to start out with, where, again, this is completely unlabeled data. There are no labels associated with these data points. And what I've done is I've run hierarchical clustering with different distance metrics, and I've stopped when there's two clusters in my data, rather two clusters in my tree. So in this first column, I'm using the single linkage method, which is that nearest neighbor distance metric. And what you can see is that if I do hierarchical clustering here, it does a pretty good job of separating out what visually should be the two clusters in this data. So all the blue points are in one cluster and all the orange points are in another cluster. If I instead use the average linkage distance metric, where again, rather than computing nearest neighbor distance, I'm computing distances between centroids of the clusters, well, my clustering here totally fails. So rather than picking up on the fact that there's two concentric rings, I'm actually just sort of grouping the top half of these together and I'm grouping the bottom half of these together. And the same thing for the complete linkage metric. So again, these hierarchical clustering methods, or any clustering method in general, is very sensitive to the type of distance that you choose. Okay, in the second row, a very similar example, where this single linkage method does a good job of separating out these two kind of crescent moon shapes, whereas these other two methods, they do okay, but they don't quite get the clusters that we might expect from them. And the reason for this is that, well, your single linkage method is very local in the sense that it only looks at sort of nearest data points, whereas something like the complete link, furthest neighbors, or average link distance metrics, kind of looking at the clusters as a whole when they compute these distances. And so they can often do strange things like, looking at data points that are far away from a particular data point when we're trying to cluster them together. And we can ignore this third row for all intents and purposes. So for an example of an actual real-world application of these hierarchical clustering methods, we saw this kind of data set before. It's a gene expression data set, where each row in this data matrix corresponds to a particular patient, and every column corresponds to a particular gene for that patient. And the colors in this matrix represent gene expression or activity. So bright green color means for a particular patient, this particular gene is turned on, it's doing lots of stuff. And a red color indicates that a particular gene is turned off. It's kind of not playing a role in their biology. And what we can do is that we can sort of cluster together genes. So in doing so, we get sort of this dendrogram on the top, where we've clustered together genes based on their activity. So genes that are very close together in this tree, very low level, are genes that tend to be activated together or deactivated together. So we've kind of found groupings of genes that behave similarly, maybe indicate some kind of biological function that's spread out across multiple genes. We, of course, can do the same thing, not on sort of a gene level, but on a patient level. So I can fit one of these dendrograms to sort of the rows in my data set. And in doing so, I cluster together patients rather than their genes. So I can ask myself, okay, are there groups of patients in my data set who have kind of similar genetic expressions? So again, these hierarchical methods are quite useful when you have some data set that you're really trying to understand, trying to get some insight about, but you don't necessarily have labels that you're trying to predict. Okay, let's briefly talk about the time complexity of hierarchical clustering. In general, it depends highly on how you exactly implement these and what distance function you choose. But in general, it's going to be somewhere between on the order of d times n squared, where d is the dimensionality of your data, number of features, and n is the number of data points that you have. And it might, on the worst case, be cubic in the number of features, or rather the number of data points that you have. So being quadratic or cubic in n is definitely not good for a machine learning problem, because often we have very large data sets. So Even if you have something on the order of a few thousand data points, this hierarchical clustering might actually be intractable. So the reason that our time complexities are as follows is that because we need to compute all pairwise distances. So there's n squared distances, i.e. We have n data points, and we need to compute n distances for each of these data points. And these are all d-dimensional distances, so every distance I compute takes on the order of d calculations. We need to do this for n minus 1 iterations, because, well, if we do n minus 1 iterations, we'll have just a single cluster at the end of the day, because we reduce the number of clusters in our data set by 1 at every step. So all in all, we'll get on the order of d times n cubed operations here. So it's dn squared for n iterations. There are smart ways to kind of get around this cubic complexity. In particular, you can save the distance calculations you do at intermediate steps, and you can get down to something like quadratic in n, but of course this is still not ideal. So what this really means is that hierarchical clustering is mostly useful for very small data sets, say less than 1,000 data points. Of course, if you have a data set with 1,000,000 data points, well, looking at one of these massive hierarchical trees is probably not going to be all that interpretable, might not even be something desirable in the first place. Okay, so let's summarize all the clustering methods we've seen so far. Clustering, again, is this idea of kind of automatically finding groups in unlabeled data, and this is an unsupervised learning technique. We've seen k-means for quite a few times at this point. This is a really simple algorithm that minimizes the squared error distance function. It kind of produces compact clusters, or rather clusters that have a linear boundary between them. And this is one of the main limitations of k-means, is this linear boundary between the clusters. We noted that it can converge to local minima and is very sensitive to initialization. So we saw that k-means ++ algorithm to help us get around that. And in terms of time complexity, it scales linearly in the number of data points that you have. We also introduced this notion of hierarchical agglomerative clustering, which is kind of a broad family of methods that we really just went through kind of the intuitive idea behind. And the main kind of appeal of using these methods is that if you're using this single link or nearest neighbor clustering, it can find non-compact clusters, or rather clusters that have nonlinear decision boundaries between them. So we saw this with that kind of example of a 2 moons data set, where we were able to actually find those kind of crescent-shaped clusters. But of course, one of the drawbacks of hierarchical clustering is this quadratic scaling in the number of data points. So really infeasible for large data sets. Okay, so with that, we will end our clustering portion of the course. So any questions at all? Okay, so we'll move on to a new topic now called dimensionality reduction. And just for a little bit of motivation, what is dimensionality reduction? Why do we care about this? So often in machine learning, you're dealing with very high dimensional data in your problems. So for example, you might have some images of faces or images in general, where the number of features you have in your data is the height of your images times the width of your images. So you might have, even on tiny images, 28 by 28 pixels, which is going to be, on the order of 1,000 pixels or 1,000 features in your data. For a text problem, you might have a bunch of text in your articles, where every word you want to consider as a feature for your article. So a long article might be on something of like 10,000 different words. Or you might be considering, say, a stock prediction problem where you have, say, all the S&P 500 stocks. In this case, you've got 500 different features to be looking at, which again is fairly high dimensional. And the motivation behind dimensionality reduction is to ask ourselves, is there some way of taking our data and describing it in a much simpler way? And what we're going to do here is learn some kind of embedding. And this embedding is a way of taking a data point and mapping it into a lower dimensional space. And the idea is that we would like to have similar data points be close to one another in this low dimensional space. And the reason this is often possible is because data often has some kind of latent, unobserved, low-dimensional structure that we can leverage to do this kind of embedding. So let's look at an example of what a dimensionality reduction problem might look like. let's say we've got a data set of hand poses. So each data point in my data set is a picture of someone's hand in a particular pose. So my individual original data points are pretty high dimensional. They're images. They've got lots and lots of pixels in them. But each of my sort of hand poses has a low dimensional latent structure to it, in the sense that I can describe each of my hand images with really only two different variables. I can describe it using the rotation of the wrist, and I can use it describing the sort of relaxation of the hand. So is it in a fist, or is it kind of an open hand? And what we can see is that if we learn some kind of embedding of these images, we can map them into a two-dimensional space rather than a 1,000-dimensional space, such that we really kind of capture the structure, the low-dimensional structure present in this data. So for instance, all of my closed fists are down here in this kind of portion, but all of my open hands are up here, and all of my not sure how to describe it, but all of my palm-facing images are on the left-hand side, and all of my sort of palm-down images are on the right-hand side. So we kind of, in some sense, take our really high-dimensional problem and have extracted the kind of meaningful structure out of it into a really nice, simple representation. This idea of embedding is really kind of everywhere in machine learning today. Okay, another cool example is in a genetic data set. So genes, of course, are extremely high dimensional. You've got thousands and thousands of genes. But what you can do is, for a bunch of patients, take their genes, some kind of rating of them, and map them into a two-dimensional space. So I kind of take this really high-dimensional vector of gene information and just project it down to just two numbers. And what's cool is you can actually see clusters in this low-dimensional representation of your genetic data. And in fact, looks like all the people from, say, Spain, all of these ES markers are kind of clustered together into one grouping down here. All of the French people are in this kind of clustering here, and so on and so forth. So this low-dimensional representation of our data is really doing a good job of kind of accurately representing sort of the high-dimensional structure of our data. And interestingly enough, the kind of way we've mapped these down into two dimensions is actually kind of mirroring the geography within Europe. So we've got Spain down here, and then France and the UK up here. So kind of a cool example of what you can do with embeddings. Okay, another way that embeddings can be useful is that they can actually tell you kind of how the sort of semantics of, say, a word might change over time. So here what I've done is I've taken a bunch of words, and I've embedded them into a two-dimensional space. So for every word, I'm assigning it a vector with two dimensions. And I'm doing this for every year between 1932 and, say, 1960, this animation will go to. And I'd like you to pay attention to the word broadcast, which is this red dot. So initially, at 1932, the word broadcast is embedded as a two-dimensional vector that's pretty close to the word, say, spread down here, and a bunch of other kind of similar words. But as I play this animation and the years go forward, well, the word broadcast eventually moves such that it gets associated with the words like radio and printed as time goes on. So the word broadcast, based on this kind of embedding visualization, we can see kind of has a change in meaning over time. So again, these dimensionality reduction techniques are really nice ways of visualizing and kind of getting insights about our data sets. And of course, yeah, broadcast becomes associated with radio as soon as radio kind of takes off in 1937. Okay, let's see a couple of techniques, or really rather one technique for doing dimensionality reduction. So let's kind of work with a really simple concrete example here, where we have a data set with two real valued features, x sub 1 and x sub 2. And here I've created a scatter plot of my data. Again, this is entirely unlabeled data. I'm not doing regression, I'm not doing classification here. It's just a scatter plot of some data. So originally, this data set requires two numbers to describe every individual data point, right? I need a number for x sub 1, and I need a number for x sub 2. My goal is going to be to somehow describe this data using only a single number z. So for every data point, I want to reduce the dimensionality from 2 down to 1. And the idea is we'll somehow use a model or some kind of function to map between this two-dimensional representation of my data and the one-dimensional representation captured by z, where f is going to be doing this kind of mapping. Well, just by looking at our data, right, there's clearly kind of a 1D structure here, right? If I just look along this direction, well, clearly my data mostly varies in this 1D direction. So one thing I could try to do is write down my model f of z such that it's a linear function of this vector v, where this vector v is really the direction along which my data is changing the most. So what I mean by that is that f of z, where z again is a single number, is just going to be z times this vector v1 comma v2. And the key here is that v is the same for all data points. So in essence, what I can do is rather than representing a data point as x1 and x2, I can represent it as this number z, which tells me kind of how far along in the direction v I'm moving. And in some sense, z times v tells us the closest point to an original data point, x1 times x2, along this direction v. What I can do after I've kind of found this direction of variation in my data is take my original data set, which again is two-dimensional, and project it down onto this one-dimensional subspace spanned by this vector v. So again, I've taken my 2D data, kind of collapsed it down into one dimension, such that my original data looks a lot like this one-dimensional representation. Really, this is the main idea behind dimensionality reduction. We like to take our high-dimensional data, project it down to lower dimensions somehow, such that this lower dimensional representation reflects the higher dimensional data. Okay, we're going to look at a technique called principal component analysis to help us do this. And the idea, again, is we're going to find the directions in the data space along which the data varies the most. So here I've got a point cloud of a bunch of unlabeled data points. And we can clearly see that there's a lot of structure to this data, right? If I move in this kind of diagonal direction, well, my data kind of follows this direction, if you like. And if I move in this other direction, it kind of captures the rest of the variation in the data. So this first direction is what's called the first principal component, maybe a little hard to see. But this first principal component is the direction of kind of maximal change or maximal variance in my data. And the second direction here is the second principal component. It's kind of the other remaining direction in which my data is changing. So really, I can summarize my data, again, using these two vectors because they capture kind of the way my data is changing. Here's an example of what PCA might look like in 3 dimensions. So I've got a point cloud of 3D data. I've got kind of 3 different vectors telling me, okay, what are kind of the directions my data is changing in? So each principal component in my data is going to be a d-dimensional vector, where d is again the dimensionality of the data. My principal components are ordered in the sense that the first principal component that I have, again this vector, is going to be the direction in the data space that really best explains the variance of the data. After I find my first principal component, I can find my second principal component, which is the direction in the data space that is orthogonal or perpendicular to the first principal component. I want it to be essentially different from my first direction, right? And best explains the remaining variance in my data. And then so on and so forth until we find all of the principal components in my data. I'll note that you can have at most d principal components, so you can have 1 for each dimension of your original data. And I'm going to assume all of my principal components are normalized so that they have length 1. So these are unit vectors. So again, my first principal component is this direction along which my data is maximally changing. And then the second principal component is the direction orthogonal to this that also captures sort of the variance in my data. So let's say that I give you a list of my principal components and I've ordered them in some kind of way. So P sub 1 is the principal component with maximal variance in my data. P sub 2 is the principal component with the second maximal variance, and so on and so forth. I haven't told you how to find these yet, but just assume I give you these directions. The way we can do dimensionality reduction with these principal components is by taking my data and projecting it onto the subspace spanned by the first few principal components. So, for example, if I have a data point x, and I'd like to project this onto the first principal component, what I can do is I take that data point x, and I take the dot product between that vector x and the first principal component. This dot product is just some scalar number, so x transpose p1 is just some real value. And I multiply that with the vector p1. So on the right-hand side of this equation here, I have a vector that is in the direction of p sub 1, because I'm just multiplying p sub 1 times some number, where this number tells me kind of how similar x is to this first principal component, or how much x varies along this direction p sub 1. So what I've done is, again, I've taken my vector x and just projected it so that it goes along this direction p sub 1. So again, we saw this example, but just to drive home what we're doing here, we've got a 2D data set originally. My first principal component is this direction in the data. So this red vector is p sub 1 here. And if I take all my data and I apply this projection, i.e. for every single data point, I do x transpose p1 and multiply it with p1, I take my data and really collapse it down to this one-dimensional subspace. And so really what I've done is I've reduced the dimensionality needed to represent my data. We really only need a single number, which is again x transpose p1, to represent every data point. Because x transpose p1, well, if we know this and we know p1, which is shared across all the data points, then I'm good to go. So I took my data and reduced it down to one dimension. Okay, if we want to project onto the first two principal components, we do essentially the same thing, where we project onto the first principal component and do the same thing for the second one. So exact same formula, we just add them together. But what we've done now is we reduced our data from needing d dimensions to represent every data point down to 2 dimensions. So we only need sort of these scalars, x transpose p1, and the second scalar, x transpose p2, to represent my data. And in general, if you want to project onto the first m principal components in your data set, it's the exact same formula, we just sum over all of the different principal components here. So, any questions on PCA? Yeah. So you don't look at individual features here. You look at an entire data point. You look at a full feature vector, and you project that onto the directions corresponding to your principal components. Okay, other questions about PCA? Okay, so I haven't told you how to actually find these principal components. And in fact, the full details of this are a bit beyond the scope of this course. If you're familiar with linear algebra, it's not too bad, but here I'll kind of just quickly go through the main idea of finding these, and don't worry about it, I won't ask you an exam question about finding principal components. But the basic steps is that we take our data matrix, X, which again, just a collection of all my unlabeled data, and I center it such that it has mean 0. So I take X and just subtract off the mean of every feature. This kind of takes my data and just places it at the origin in the data space. The next step is to compute the so-called covariance matrix of my data, which in this case is going to just be this matrix product of x transpose times x. So this matrix C here is a D by D matrix. It's got one row per dimension and one column per dimension. And the entry C sub ij is the covariance between the ith feature and the jth feature, where really the covariance tells us how much a particular feature kind of varies with another feature. So how similar are these two features? To find the principal components in my data set, what I need to do is find the eigenvectors of this data matrix C. So again, if you've never seen an eigenvector, don't worry about it. But there's a lot of numerical routines out there for finding eigenvectors of a given matrix. So overall, we can find principal components in just 3 steps, but kind of the math behind these is a little bit beyond the scope of this class. In terms of code, not too hard to implement this, maybe 3 or 4 lines. So mu here is the mean of all my features. X0, I'm centering my data matrix, so I take my data matrix and subtract off that mean. S here is just x transpose times x, so this is my code.